Good afternoon, everybody. I would like to welcome you in the 7th seminar of the HIAAD. Today, Nikos, Professor Nikos Scarmeas will be the chair of this very interesting and very clinical oriented, but with a very broad approaches seminar. And we hope you will have a lot of questions. So Nikos, the floor is yours. Thanks again for organizing this very interesting meeting.
Okay, thanks. Thanks, Yanni. So hi, everybody. So the idea behind these HIAAD seminars is to try and be as translational as possible. So today they're slightly different in that they do not involve much of basic research aspect, much of laboratory aspect, but they have a very strong research component. So there's a clinical and a research component, but not particularly lab one, let's say laboratory one. So in any case, we'll talk about new polygenic risk scores, and it's a recent method, and there has been exponentially increasing literature on this. So we'll have, I think, two very interesting presentations. In the beginning by Dimitris Avramopoulos. So Dimitris is a professor of genetic medicine at Johns Hopkins in Baltimore, in the States. And then Dimitris' presentation will be followed by a presentation by Alex Hatzimanolis, who is assistant professor at medical school, assistant professor of bioinformatics at medical school here at the National Kapodistrian University of Athens. The idea is that Dimitris may talk to us a little bit more about summarizing the concept of PRS, how they are constructed based on GWAS data, and provide a more general framework. If there are slight deviations from this, it's fine. And then Alexandros could talk maybe a little bit more about dementia-related PRS, potential clinical uses, maybe a few things about their relation with potential underlying biology and so forth. So without further delay, Dimitri, the floor is yours. Just a reminder, the idea is to have short presentations, like 15 or 20 minutes each, and then maybe have some questions. But so that's an approximate, time frame. Dimitri.
I apologize for that. I was muted. Let me. Can everybody see my screen now?
All is fine, Dimitri, but we can see your screen and we can hear you.
Thank you. So I will kick this off. First of all, thank you, Nico, for the introduction, and thank you for inviting me to participate in this seminar. I will kick this off by giving some general concepts and methodologies, as Nikos mentioned. And I can't really talk about PRS scores unless I explain a little bit about diseases with complex genetics. So most common phenotypes, whether they are diseases or whether they are normal phenotypes like height, blood pressure, etc. So most common phenotypes follow complex genetics, which means that as opposed to Mendelian disorder, where one genetic variant can determine whether one gets sick or not, there is multiple genetic variants involved that increase the risk. And those multiple variants also involve multiple genes. Most of them, however, are not, unlike Mendelian genetics, sufficient to cause disease. And while interactions have not been completely ruled out, it is accepted that they mostly affect the risk in an additive way, independent of each other. And having mentioned that, I have to give credit to Ronald Fisher, who more than 100 years ago today was able to reconcile Mendelian and biometrical genetics, biometrical being continuous phenotype like height, and was able to show how you can actually have a genetic effect that goes stepwise with added effects of multiple genes. Until then, it was thought that you either have a mutation that causes a disease or you don't. It was unclear how one can have a continuous phenotype. So the first attempts to map complex diseases like Alzheimer's disease, but also like many other disorders, cardiovascular disease, schizophrenia, etc. The first attempts were done by genetic linkage, as was done with Mendelian disorders to that day. And people were looking for large pedigrees in order to that segregated the disease in order to map the genes. And that was in fact successful for Alzheimer's disease with the discovery of presenilin 1, which also led through its similarity to the discovery of presenilin 2, as well as the APP gene. And these all together led to the amyloid hypothesis for Alzheimer's disease, which is now becoming more and more clear that it's not the whole picture. However, these mutations, these variants that cause Mendelian disease are less, can account for less than 5% of Alzheimer's disease cases. So this was followed, so soon it was realized that linkage will not work because we don't know what is called the parameters of parametric linkage, which is how much does the variant increase the risk, and how often is it that one can have a disease without having a specific variant. So other approaches were tried, like non-parametric genetic linkage, which is mostly mostly based on allele sharing between diseased individuals, but that was not very successful, in fact, not for any disease. And even in Alzheimer's, although there was a weak signal over the ApoE locus, it never became strong enough, and it never showed any other loci that were responsible. At the same time, people tried genetic association, which is the approach where you compare the frequency of specific alleles that can cause disease between patients and control individuals, and you're looking for differences in their frequency. And that's how ApoE was identified in 1996. I'm sorry, 93, I believe it was. The problem was, though, that what we didn't know at the time was that ApoE was the exception and not the rule, having a very large effect on the risk. And this ended up with people performing lots of different tests for different candidate genes and reporting, due to publication biases, many results that were not actually valid. And this, however, it was through a landmark paper in 1996, Neil Rees and Kathleen Marikangas showed that although there's limitations in the power of association studies as well, and the reason for those limitations is because it requires genotyping millions of variants, and you have to pay a large statistical penalty, which means that you need to achieve very low significance p-values. So although there are limitations, their power is actually very much superior to linkage. And on the table on the right, in red, they show the number of individuals or families required to identify genes with different effect sizes. You'll see that the smallest effect size and odds ratio 1.5 that they tested, it would require millions of pedigrees, while when one used association, the numbers were much smaller. What they didn't know at the time is that actually even this odds ratio of 1.5 is really significantly above what is actually the true underlying effect sizes. So then this was, although this at first seemed impossible because the numbers were big and because the numbers of genotypes we needed were big and the technologies were not there, this did start the era of GWAS, where technology quickly allowed genotyping hundreds of thousands of single nucleotide polymorphisms at the same time. And at the same time, sample sizes grew to sizes of many thousands through large collaborative efforts. So as a result, additionally stringent criteria were set for reporting to avoid false positives. And as a result, in the 2000s, we had the blooming era of GWAS with thousands of variants discovered robustly associated with hundreds of phenotypes. And it was observed that most of them do not change proteins, but rather most of them are in gene regulatory region and likely change the expression levels of genes. In this review by Manolio et al., by Terry Manolio, she describes what the genetic landscape of disease is in general. And you see on the x-axis, she has allele frequency of different genetic variants, and on the y-axis, the effect size. And what she says that when it comes to large effect sizes and common variants, there's very few examples. One of them is ApoE, where you see for a allele frequency of around 15%, down here somewhere, it has an effect size of 2.5, so it falls somewhere around here. Then there is the more common scenario where you either have common variants but that have very small effect, mostly less than 1.5, or intermediate variants that have pretty low frequency, less than 1%, but also have a very low effect size. And these are often found by sequencing studies today. Or you have very, very rare variants with frequencies of less than 1 in 1,000 or less than 1 in 10,000 that have very high effect size, and those cause Mendelian disorders. And of course, theoretically, there are also rare variants that have a very small effect, but because of their rarity and because of their small effect, they're practically impossible to experimentally identify, although they might actually account for a significant portion of the heritability of the disease. So, as I said, this was in the 2000s was the beginning of the GWAS era, and this is an example of a genome-wide association study for schizophrenia, which was one of the diseases that had the most quickly accumulating sample sizes. And in this study, they looked at 3.3 thousand European individuals with schizophrenia and 3,500 controls. And their results were actually quite disappointing. So in the figure on the left, you see the most significant finding, which was a SNP in the HLA region, which barely reached statistical significance. However, having spent all that time and effort to collect samples and all that money to genotype them at the time, which was far more expensive, they started to look at other ways to look at the data, and they rationalized that, yes, all those things that are not highly significant with p-values less than 5 to the 10 to the minus 8, those that are significant to a degree, let's say 10 to the minus 6, are still very likely to be true positives, although one cannot formally claim that. And so those that are at this level are likely to be enriched in true positives. So maybe we can use the effect size that we have calculated to generate a risk. And this is the first time that the polygenic risk scores were reported in this way. In this study, we showed that when they generated based on their GWAS a polygenic risk score, and then they applied it to other studies, other schizophrenia studies, they always found a significant correlation between disease and the polygenic risk score, validating that this actually works. You see here that in the middle of the 3 schizophrenia samples, there's an African American sample which also shows significant correlation, but very much reduced. And this is because the ethnicity is very important to be kept the same between the reference GWAS and the target GWAS, as I will show later how that works. And interestingly, they also found that they could actually correlate this polygenic risk score for schizophrenia with the presence of bipolar disease, which suggests that there's an overlap between the two psychiatric disorders. And in fact, as I will show later, the overlap extends beyond that. So how is this polygenic risk score calculated? So the PRS is calculated by the sum of an individual's risk alleles weighted by the risk allele effect size as derived by GWAS data. So here's what happens. One takes what is called the reference GWAS, which the larger the better, so that the data are more likely to be valid, and sets a threshold of p-value. I have an example here of p less than 0.001, which is by no means genome-wide significant. However, it is some of the top significant variants, and there's still going to be tens of thousands of them. And for each SNP, takes the beta, the effect size that's calculated by the GWAS, together with the allele that's found to be the risk allele, and goes to a target sample. And here we see one single individual of the target sample with the same genetic variants and the SNP name here, and the number of alleles they have, the number of risk alleles they have for that variant. So based on that, a risk from that SNP is calculated, and because, as I mentioned, these are additive, one can add them all up and find the overall the polygenic risk score at this p-value threshold. Now, as I mentioned, some of these SNPs are true, some of them are not. So there's a signal and there is noise in the data. And by setting different p-value thresholds, one can change the signal to noise ratio. And because we don't a priori know what the best threshold is, people are doing multiple thresholds, as I showed in the previous slides. So what you see here is the different green bars with different shades show results at different p-value thresholds, and some of them are highest at different places. So if you want a lot of detail about how to perform a polygenic risk score, there's a very nice paper in Nature Protocols, 3 years old now, that talks about everything you need to know about how to quality control the data, how to adjust for linkage disequilibrium, which is a correlation between genetic variants in the genome, and how to perform testing and validation. Further, as I mentioned before, the overlap of schizophrenia with bipolar disorder. There's been a lot of work on schizophrenia for this. So that shows overlap with multiple psychiatric disorders. There has not been as much work in Alzheimer's disease. There's a paper showing that the PRS for Alzheimer's disease correlates with amyloid load in the brain in individuals without an AD diagnosis. But I think there's a lot more to be explored there. The clinical utility of polygenic risk score becomes really more and more obvious as people look at larger samples. So in diseases like cardiovascular disease, atrial fibrillation, and type 2 diabetes, where very large samples have been accumulated, one can predict how many individuals, what percent of the samples have more than a 3-fold higher risk based on polygenic risk score alone. So that's an important clinical information to have when you're looking at the risk for those disorders. In post-mortem diagnosed sporadic early onset AD, the predictive ability of PRS has been found to be between 65 and 75%, which is a fairly good result. And as it has been shown in cardiovascular disease again, combining this information with cognitive scores, the data on brain atrophy with any kind of clinical information can greatly improve the utility of PRS in the clinic. And as GWAS are getting larger and larger, and the signal to noise becomes stronger and stronger, I think that in the future the contribution will be even greater. Now, there's other ways to use polygenic risk score, and I have here an example for my family collected here in Greece in collaboration with Alexandros, with Nikos and other names you might recognize in this paper. This was work on schizophrenia, where we are doing linkage in a large pedigree with multiple affected members to identify genes genes that segregate mutations. And in fact, we identified a gene called Caskin-1 that carried a mutation that segregated with the disease, and we also showed that it is functional. However, in this family, there were two individuals that, although they had the variant, number 3 and number 5, although number 3 in the first generation and 5 in the second generation, so although they had the variant, they did not have the disease. Now, this is not uncommon in Mendelian genetics, but we wonder if that has anything to do with overall genetic risk. So we run polygenic risk score analysis, and the results are on the right, where the schizophrenia polygenic risk score we see is higher for all people that actually did get affected, had the mutation and did get affected, and the lowest two are the ones that did not get affected. Our group has also recently published, together with Vaso, information showing that there's possibility that we can improve the utility of polygenic risk score by including additional functional information. And this is again an attempt to improve the signal-to-noise ratio. So instead of looking at all variants of a specific p-value, we can weight them, or we can only look at those that have functional evidence. Which brings me to alternative approaches to polygenic risk scores. As I said, the GWAS have had many results, and in Alzheimer's disease they have shown many pathways involved. And it is more than likely that different pathways might drive the disease more in one patient versus another, which could be very useful in precision medicine. So it would be interesting to filter variants depending on which pathway they contribute to the risk to. How does one do this? Well, first you have to link SNPs to genes. Some people do this by location. This is not a good approach in my view, because most often SNPs do not influence the gene that they are located nearest to. Like 50% of the time they influence some genes further away. I think that the most fruitful approach is to do it by EQTL, by expression quantitative trait analysis, meaning filter for SNPs that actually look at only SNPs that actually change the expression of a gene, and those can be found in databases like GTEx. The second step is to subset the genes based on the pathway they belong to. And again, there's a number of the databases where one can do this. And once you have these 2 points of information, you put them together and you can filter your SNPs to only include in a PRS those that change the expression of a gene, and that gene belongs to a specific pathway. So we have done some of that work in collaboration with Gwen Smith and Johns Hopkins, and I'm just showing one example of this work. So Gwen had amyloid PET data, and she showed on the left many different brain regions that she had data on. And when we do a polygenic risk score with all the SNPs, you see in green the correlation, the R square of the polygenic risk score with the amyloid accumulation in different regions, and in red the p-value. Those are almost all significant. But when we subset to genes that have any relation to amyloid production in general, then we see that in certain regions the correlations increase in some cases quite a bit and become significant when they were not, despite the fact that we have reduced the number of genes considerably. And interesting, when we look at inflammation genes, we see in certain, like in the amygdala in this case, or in the thalamus, we see the correlation becoming much, much stronger, again, despite the reduction in the number of SNPs. And on the right side, you see the significance of the polygenic risk score, and one example from all this data and how it's done on many different thresholds. So I will close here just reminding you what the advantages of polygenic risk scores are. For one thing, they leverage the power of large GWAS, which means that we're using cutting-edge information to characterize the disease. They avoid multiple testing, because once you have one score, then you do just one test, as opposed to testing multiple SNPs for one phenotype. They allow comparison of the genetics of different diseases and comorbidities, like the example I said in schizophrenia and bipolar disorder. They allow the dissection of disease mechanisms through pathway-specific polygenic risk scores and the attribution of different disease features. And by doing so, they can be very valuable for precision medicine, allowing genetic profile-based clustering of patients and maybe an individualized treatment or prevention. And I will stop here. Thank you.
Okay, Dimitri, thank you very much. Very nice journey from linkage analysis to association studies, and then talking to us a little bit how we can maybe increase our prediction regarding disease probability using PRS, probably in conjunction with the clinical information, but also how PRS can maybe be an explanation for variable penetrance in monogenic diseases like the schizophrenia example you showed us, and also how pathway-specific PRS analysis can help us maybe fine-tune or improve the PRS scores that are currently being constructed. So thank you very much. So now we'll proceed with Alex. Please, Alex, go on. The floor is yours.
Sure. Trying to upload my slides. You can see the file here.
Do you need any assistance in sharing your screen?
I think I am Zoom. It's not. I mean, the platform does not recognize Zoom, so I have to give access to Zoom first, I guess.
Okay, please let us know if you need any help.
Just give me a minute. I think I can solve this.
I was wondering whether we could have any questions while giving some time to Alex to resolve the technical issues. Does anybody have any brief comments or brief questions for Dimitri? Maybe Yanni, you wanted to ask something?
Yes. I mean, you wanted to say something and I interrupted you, Nico?
No. I was wondering whether you had a question. That's what I was asking,
Okay.
Giving some flexibility for Alex to load. Yeah.
Thank you very much for this very enlightening presentation. So I was wondering about how you can account for the effect of ethnicity or let's say population background for the polygenic risk score, both from the part of the GWAS where it is derived from and from the part of the population it is applied to. Can you account for different populations?
Yes, the problem here.
Sorry.
Almost in the Caucasian is changing, and now there's more GWAS available from Africa. Can you hear me?
I think it's making interruptions, but I'm not sure.
Yeah, there's some. Maybe you can try again, Dimitri,
Okay.
because it was in the.
Yeah, I was saying that one problem is the reference GWAS that are available. This is most of the problem. One can use statistical techniques to correct for stratification, but the power is always reduced when you don't use the same reference and target. This is not so because the most GWAS are from European populations. This, although admixture still exists, of course, within Europe, it's not so much of a concern in Greek samples, although it's best if one accounts for it. And things are getting better for African samples and Asian samples. So again, it's a matter of the problem is power. Power will be reduced, and there are corrections, but they never overcome this problem. And the best we could do is do a large GWAS in the Greek population, but that does require effort and money.
And can, if I may go on, can you derive the PRS from exome data, where in some cases maybe you have some missing values from some SNPs, or is it easy to derive the PRS from exome data?
I know that it is possible. I don't know if I would call it easy. You can use, but you can use. You can also, for one thing, you'll get less information for the SNPs that you're missing, but you can also impute some SNPs and get more information that way. So you can certainly do it, but your power might be decreased a little bit.
Okay, great. Did we resolve the technical problems?
I'm afraid not, and I'm not sure why.
Alex, do you want to send your slides and somebody else can share them for you?
Yeah. It doesn't allow you.
Yes, but I think if we do that, we cannot change the.
If you send them,
slides.
well, you can say next, and you could send them to any of us maybe, and we can share. And then if you say next, we may be able to change it. It doesn't allow you to share?
I can, well, everything in my computer says I can't share, and I don't understand why. I mean, that's very
stuff. Do you think one could look at whether variation in the way the PRS was constructed might strengthen or weaken those two associations? Because one of the questions I have is, are amyloid and inflammation part of the same pathogenic cascade? Right. Do they vary by people, as might be reflected then in a differently nuanced PRS?
Yeah, so this is a lot of the work that I want to be doing now. There's many parameters that one can change. For example, one can change how strong you require the relationship between a gene and the specific pathway to be. In what I showed, I was pretty relaxed, but maybe if I get more stringent, maybe it'll get better, or maybe it will get worse. I can't know that beforehand. There's that. There's also, as you said, we can look at the intersection between different. We could look at inflammation and amyloid, or we can look at the union, inflammation or amyloid. So it will take some exploratory work to find out what is best, what works best. There's also other parameters, such as I'm saying I want them to be correlated with the expression of the gene, but this correlation is associated with a p-value, and I need to set a p-value threshold. I had set for this work as a genome-wide significance threshold, which might be too stringent. Maybe I should lower that, because maybe I'm throwing away a lot of useful information. So all this needs to be explored.
Okay, great. I think Alex has been able to resolve the technical issues, so maybe we can proceed with.
Okay, I'll start so we can speed up things a bit. I'm going. Thanks first of all for the invitation to participate in this seminar. I'm going to be talking about recent findings from large-scale genomic studies in Alzheimer's disease, and I'll show you results related to polygenic risk scores for Alzheimer's disease, which might be of potentially of clinical relevance. So Alzheimer's disease, it's the most frequent type of dementia in older population. AD has a significant heritability component, which might contribute up to 60 to 80% of its heritability. And it is important to note that a significant proportion of the heritability of AD can be explained by common genetic variants across the entire human genome. And the estimation of the amount of heritability explained by common SNPs could be up to 53% of heritability. This is the latest genome-wide association study for Alzheimer's disease. This was reported about a year ago. So this is the Manhattan plot. It shows the association results for every common SNP in the genome. This study included, as you see here, about 100,000 AD cases and almost 700,000 healthy controls. 83 genomic regions were robustly associated with the development of AD. And of course, up here on chromosome 19, you can see the well-established ApoE risk locus for Alzheimer's, which showed a very statistically significant signal here. From the GWAS data, as Dr. Avramopoulos mentioned, we can construct PRS scores from Alzheimer's disease. As you see here, these scores have a normal distribution in the population, and we, as you see, we can discriminate AD cases from healthy controls with a fairly good accuracy, and from large samples, large population genetic studies, the area under the curve, an estimation of how good is the predictive ability of PRS has been estimated at 75% for clinical samples and 84% for pathologically confirmed samples. So the PRS accurate predictive ability for Alzheimer's disease is much higher than compared to other complex neurological and psychiatric disorders. Excuse me. In the same GWAS study from the European Dementia Consortium, it is they also found that, as it is shown here, that when we stratify the population to high and low scores according to PRS score, and estimate the progression from the healthy state to AD over time, individuals belonging to the highest decile of the distribution of the PRS distribution develop AD earlier. And similarly, in another study, they found a similar result, that the high PRS scores may progress more rapidly from mild cognitive impairment, MCI, to AD. In another large population study, this is the data set from the UK Biobank. they have computed the PRS score for AD for their population, almost 40,000 people. And an interesting finding here was that more AD cases were found in the upper distribution, the upper quartile of the distribution of PRS score. That's 2% of those individuals, compared to a very low percentage, 0.1% of AD cases found in the lower quartile of the distribution. So it becomes clear here that PRS might be a valid genetic risk factor for AD in the population level. Another example, a third study, it's from the ASPRE study, almost 12,000 individuals examined in this study with an age over 70 years, and DSM-4 based diagnosis for dementia. And here we can see again that PRS computed with different methodologies, computational methodologies. We see here that again the individuals with belonging to the 10th, the last decile of the distribution, compared to those in the first decile, have an increased odds of developing dementia. So in more detail here, when we compare these two groups of people following stratification by PRS, they found a threefold higher risk of incident dementia with one methodology when they compare only 83 genome-wide significant genetic variants, and almost fourfold higher risk for dementia when they examined the entire number of SNP across the genome. Another study by the European Dementia Biobank Consortium showed the results following stratifications by PRS and, of course, ApoE genotype status. As you see here, we can see that when PRS, of course, is significantly associated with higher AD incidence, but when we stratified by ApoE E4 allele status, again, we see that homozygotes for E4 AD risk allele and a high PRS score for Alzheimer's disease had an increased risk for developing dementia. And the same study, they also reported that this particular group of individuals showed an earlier median age at onset of the disease, which of course may have a significant clinical importance. So I will turn to our own research work in Genedis Hospital, and then I will present some very interesting results from our work, from associations between PRS scores for related to AD and dementia, with multiple phenotypic outcomes in the HIAAD study, which is coordinated by Professor Scarmeas. A few things about this population sample, the HIAAD. This is a sample of almost 2,000 older individuals enrolled in HIAAD and recruited from two major cities in Greece, Athens and Larissa. Multiple assessments were made with regard to dementia, AD incidence, MCI, cognitive performance and decline, frailty, and of course, socio-demographic data, assessments related to nutrition and lifestyle activities, as well as neuropsychiatric symptoms. And thankfully, this the HIAAD sample was genotyped, was part of the European Alzheimer's and Dementia Biobank Consortium, and it was genotyped. Almost 1,400 individuals from HIAAD sample was genotyped across the genome, and we calculate polygenic risk scores for later investigations with the phenotypes I mentioned a while ago. These are a bunch of recent papers reported from our group in Genetium. Let's see a few. Let's describe some of our findings so far. So this is a study investigating a vascular burden score for dementia together with the AD-PRS score. Firstly, it was shown that a sum score of clinical cardiovascular risk factors for dementia was associated with global cognitive performance in HIAAD sample, as well as increased prevalence of dementia, as you see here. And when we combined this information with PRS score for Alzheimer's disease, higher vascular risk score showed worse cognitive performance across all 4 AD-PRS tertiles in the sample, but multivariate analysis did not actually show evidence for an additive interaction between the clinical vascular burden score and the AD-PRS. In another study, we examined how AD-PRS might moderate the adherence to the Mediterranean diet in AD risk, and as you see here in the left, but individuals with low PRS, with AD-PRS, and low adherence to Mediterranean diet had a almost 10-fold higher risk of developing AD per year of follow-up. Just to mention that these individuals were followed up for 3 years, and assessment for cognitive function and AD incidence were assessed at 3 years follow-up. So yes, here we see that Mediterranean adherence to the Mediterranean diet was a risk factor in participants with a low risk, genetic risk for Alzheimer's. And here a more biologically informative PRS. We constructed the PRS score for beta amyloid levels, and we checked for associations with AD and MCI incidence in the sample. And as you can see here, higher PRS for beta amyloid levels, as well as cognitive reserve, were associated with a 34 higher risk and 8% less risk for AD or MCI incidence, respectively. In this case, an additive interaction was observed between a PRS score for beta amyloid deposits and cognitive reserve was observed, as you see here. low PRS for beta amyloid and higher cognitive reserve. Could be protective factors for AD or MCI incidence with 63% less risk. And this is our latest results, given that inflammatory brain dysfunction has been implicated in the AD pathophysiology. We constructed polygenic risk scores for interleukin 6 levels. And as you can see here, we observed that genetic predisposition for higher polygenic risk for interleukin 6 was associated with greater AD or MCI incidence. And just a few concluding remarks to wrap up. Up to 50% of Alzheimer's disease heritability might be explained by genetic effects due to a large number of common genetic variants. Polygenic risk for AD is implicated to increase incidence of dementia and an earlier age at onset in population-based cohorts of older adults. Higher AD predicts worse cognitive performance in older adults independently of cardiovascular clinical risk factors. That's the example with the vascular burden score I mentioned before. AD-PRS may influence the relationship between adherence to the Mediterranean diet and AD risk in non-demented individuals. And lastly, an additive interaction between polyrisk for elevated beta amyloid levels and lower cognitive reserve associated with higher AD or MCI incidence. And thank you for your attention.
Oh, thank you very much, Alex. We are already
quick enough.
Yeah. So maybe you can stop sharing.
All right.
So it's already at the end of the hour, and because of these technical issues, we're a little bit running behind. Not so much, but I'm not sure whether Yanni will have some time for maybe 5 minutes for questions, or whether we should wrap it up.
We usually have, especially because of these technical issues. So if there are questions, I have one, but please.
Yeah, sure, please go on. You represent the basic science community, so
very fast, very pointed. What is your comment, or Nikos and Dimitri could also comment, but what is your comment on the last data where amnesic MCI and AD with the higher polygenic risk score were the same based on the inflammation? So do you think that we're talking about common mechanisms in the way between the MCI, this amnesic MCI, and the AD you are checking through the inflammation, and how the polygenic risk score could push and connect these two categories? Or is it possible that this MCI, you characterize the patients as MCI, could already have started Alzheimer's disease, but we didn't reach the clinical criteria to enter the AD diagnosis? So I'm talking about also the way we are collecting samples for biological biomarkers or whatever. This is a very critical question, whether we should separate MCI from the very beginning based on the diagnosis, or we follow a more, let's say, data-driven or clustering independently of the diagnosis, but based on other biochemical, clinical, whatever data. So what's your view on that? I know it's a big question, big issue.
Well, I'm not a clinician, so I can't comment on the clinical part of differences with regard to differences between MCI and AD, but I would say that the polygenic risk for inflammatory biomarkers could be a sign or a predisposition, a biological predisposition to MCI, which might give rise to AD pathology. But it depends on other factors too. It's not only genetic.
Your clustering were together. You put together MCI/AD. That's
Yeah,
what.
that's in our analysis, because the sample size is limited and we don't have enough power due to the limited number of AD cases. So we try to combine these two groups, MCI and AD cases. I think it is logical to expect a common biological predisposition in both states, but still, I can't make any clinical argument here. I mean, this is. I think Nikos might be.
Yeah. I can say very briefly. So I wouldn't focus very much on the distinction between MCI and AD dementia. It's really different stages. And we focused on amnestic and AD so that we have some relative underlying homogeneity. But I think the main point here is that, we haven't seen very much in the literature, in previous literature, relating, predisposition to inflammation through genetic instruments and risk for clinical dementia, mild or more severe. I hope we haven't missed much. So it's an interesting aspect. And I guess one way to explore different mechanisms is the pathway-specific PRS fine-tuning that Dimitri described before. So where you have a PRS for a clinical outcome, a PRS for dementia, okay, and then you can, see what pathways are involved. Another way would be to construct PRS for different underlying biological pathways like inflammation here, or like the amyloid and so forth. And in theory, with the precision medicine approach in mind, one could think of combining different PRSs for different underlying mechanisms, no matter how they are constructed. But I think the methodology is still something that is being developed on that end. So the idea would be you have a person and you have a certain predisposition for inflammation and some other predisposition for tau or for amyloid or for vascular or for oxidative stress. And maybe somehow we could sum these and create a more general genetic risk score. I don't know if Dimitri has any comment on that.
No, the only thing I wanted to add to all this is that, you could have two different clinical diagnoses. Actually, in fact, two very different clinical diagnoses like schizophrenia and bipolar disorder that I mentioned earlier, that could have very strong common genetic backgrounds. So in that sense, by combining them, what you do is that you're distilling to the commonalities while you might be losing some of the things that are not common to each other. But I don't think there's anything wrong with doing that, as long as you're aware of what you're doing.
Okay, great. Any other questions, or do we have any questions from the chat? I couldn't see anything, or from people who are watching through YouTube, or anybody else from people who are
I think Yannis Zaganas has one.
Yannis.
I hope I don't take too much time over the hour. So it's very interesting, and I must say it's very impressive, these results. The only thing I would think about is maybe in extension to what Nikos Scarmeas said before, maybe you could try to see if you can do some kind of gene-based analysis. So take separate genes and see how they interact between them. And another comment that I think we're losing, but I'm not sure how much we're losing, is rare variants, functional rare variants that are not, identified by this methodology. So what would you think would be the percentage of heritability, or let's say risk conferred by rare variants not accounted for by the PRS?
Well, I can comment on that. As I showed in my first slide, the proportion of heritability due to common variants is between 20% and 50%. There is still room for other genetic effects. So yes, I agree that rare variants, copy number variants, other kind of genetic other kind of genetic changes in the genome might contribute to the heritability of dementia.
And I don't know if you remember that I showed this slide that showed allele frequency versus effect size. And there was the bottom left corner, the variants with low contribution, with low effect size, but also with low frequency. And although individually they have low frequency, there are tens of millions of them. So altogether, they account for a significant chunk of the heritability. I would predict that we'll never really know them all, but we can always keep improving. There's always space for improvement, and it's all a matter of cost, really, as technologies make it easier. And maybe as, I wouldn't be surprised if at some point every human born, and that depends on politics in the country, but if every human born gets sequenced at birth, at some point we're going to have data on the entire population, and we can find things like that, and we can have much better predictive value. Of course, this sounds a little bit like Big Brother as well, so there's a lot of things to be discussed on this. But yeah, I think the possibility is there.
Looks terrifying.
Yeah, great.
Somehow
So,
terrifying.
yeah, we're 10 minutes after the hour, so I think if we don't have any other questions, I just wanted to add that, you know, we are in a process of collaborating, you know, from Athens with Dimitri, you know, within the HIAAD framework. It's one of the collaborative projects, and try to
No, yeah, thanks. We will have our next meeting. We will not have in August, and we will have probably in September. You will receive in advance, on time in advance, the announcement about this seminar. So there will be no seminar for August. We would like to wish everybody happy holidays and enjoy and rest. And we will see each other in September with, again, both clinical and basic research bridges in HIAAD. Thanks a lot for everybody. Thanks, Nikos, for organizing this great. Thanks, Alex and Dimitri.
Thank you.
Thank you.
Bye.
Bye-bye.
okay. Bye, everybody.
Bye.
Bye.
All. Bye.